We're going to do a quick review of an action potential and then move on. So remember, it starts with a graded potential. That is, there's a stimulus and the ions switch places across the membrane. Positive ions enter the cell. And if there is enough of a stimulus, the threshold potential is reached and an action potential is triggered. So there are 3 steps involved here. The neurotransmitter is released onto the cell body and opens the sodium channels, causing a larger depolarization. The sodium channels close and potassium channels open, causing repolarization. And the sodium-potassium pump resets the original ion balance. So here our sodium channels open. Sodium rushes in, then potassium channels open, potassium rushes out, and repolarization occurs. Well, there's 2 ways that an action potential can flow down an axon. One is called continuous propagation. That is, it starts and goes step by step along the axon, sarcolemma, neurolemma, until it reaches the end. But if an axon is myelinated, what actually happens, it is as though the action potential jumps from node to node. The node is the bare axon in between the myelin sheath cells. So that is where the action takes place, at those nodes. And so saltatory propagation is much, much faster. Therefore, myelinated axons carry faster axon potentials. You might want to pause here to read through this diagram. So in terms of speed, larger diameter axons and myelinated axons will carry the fastest signals. So therefore, urgent information is carried along large diameter myelinated axons. Which ion flows into a neuron to depolarize it? Sodium. The action potential will travel along the axon, and there is a transfer of the action potential from one neuron to the second neuron or to an effector at the synapse. The presynaptic cell axon forms axon terminals that contain neurotransmitters in synaptic vesicles. The narrow space between the presynaptic membrane and the postsynaptic membrane is called the synaptic cleft. You probably remember much of this from the neuromuscular junction. But we'll review it anyway. So step 1, the action potential arrives at the axon terminal and depolarizes the synaptic terminal. Extracellular calcium enters and causes the release of acetylcholine. Acetylcholine crosses the synaptic cleft and binds to receptors on the second cell. This causes sodium to rush into the cell. The acetylcholine that does this is removed by acetylcholine esterase, the enzyme. Over 100 different neurotransmitters exist. They all work in different ways. We've already talked about acetylcholine, which is found at the neuromuscular junction, but elsewhere in the body as well. And here are some of these neurotransmitters: epinephrine and norepinephrine. Serotonin, you've probably heard of. Serotonin is important in our emotions, and it is the serotonin receptor that that illegal drugs target. And you get a pleasurable feeling from those illegal drugs because they are producing a larger effect than serotonin, but similar to serotonin. And glutamine is inhibitory. Where would serotonin most be concentrated within a neuron? Well, it's a neurotransmitter, so where would it be? Within the synaptic vesicles at the axon terminal. The second part of this chapter is talking about the central nervous system, spinal cord, and brain. Most of the body's neurotissues in the spinal cord and brain, and there is no relationship between brain size and intelligence. You do not have to know the development of the brain, but here's a 4-week-old brain. Well, our brain is divided into 2 hemispheres. This brain is mostly white matter, but it's covered by what I call the icing, an icing of gray matter. And that gray matter is the cerebral cortex. And that gray matter is really, really important for us. Remember, white matter is myelinated, so it's for speed. It is the gray matter, the cerebral cortex, that functions in thinking and processing and memory and regulation. And of this brain, there are certain surface features you need to know, and those features are fissures, gyri, and sulci. And we'll see a picture of that in a second. The cerebellum is the second largest structure in the brain, and that coordinates our muscle commands. The diencephalon it consists of the thalamus and the hypothalamus. The hypothalamus we saw in chapter 1, it's important for maintaining homeostasis. It's a center for emotions, automatic, that's that word, autonomic functions, hormone production. But the thalamus is a relay center for incoming sensory information. From the spinal cord going into the brain, this is called the brainstem, and it consists of 3 parts. First, the medulla oblongata, then the pons, and then deep into the brain will be the midbrain. All of these are relaying sensory information into the brain. So the medulla oblongata controls heart rate and blood pressure. I would say just stick with the fact that the brainstem maintains autonomic functions. There's some deeper functions of heart rate, blood rate, some control, and there's a part of the midbrain that helps us maintain consciousness. So here's a picture of the brain. We're looking at it from the left side. This is the anterior of the brain. In the posterior portion is the cerebellum, or little brain. Here's the spinal cord coming up into the brain. So the bone would be right here, just right outside the cerebellum. And remember that spinal cord goes through the foramen magnum. So right here where it enters into the skull will be the brainstem. And here's a close-up of the brainstem where we have the medulla oblongata, the pons, and the midbrain. And just above that, or just superior to the midbrain, will be the diencephalon that contains the thalamus and the hypothalamus. Now, this is the left cerebrum, or the left cerebral hemisphere. And you can see it consists of these convolution called gyri and sort of shallow folds called sulci and deeper folds called fissure. For example, look at this deep, deep fold here, and that is the lateral fissure. Now, the lobes of the brain are named that have the same name as the bones outside the brain. So this would be the temporal lobe, frontal lobe, parietal lobe, and occipital lobe. Inside the brain are chambers, and the chambers are lined with neuroglia called ependymal cells that produce cerebral spinal fluid. And you can see the odd shape of these blue ventricles. You don't need to know any of these terms here, but this, the ependymal cells are lining these ventricles and going down through the spinal cord. And so the cerebral spinal fluid is produced and travels up and down the spinal cord and around the brain. The corpus callosum is something that you would see if you were dissecting the brain. if you pull the two cerebral hemispheres apart, you're going to see white matter connecting them. So it connects the information between the left and right side of the brain. Also, inside the brain, you're going to find groups of cell bodies. Now, cell bodies are part of gray matter, so it'll be clumps of gray matter. These are called nuclei. There are basal nuclei in each hemisphere, and they help direct some of our movements. So this is a coronal cut of the brain. We've cut into the cerebellum. You can see this structure of white matter in the cerebellum. This is called the arborvitae. But you can see the cerebellum is coated with an icing of gray matter, just as the cerebrum is coated with an icing of gray matter. Here's some clumps of gray matter, basal nuclei. All this blue here is part of the ventricles. This here is connecting the left and right hemisphere. It's the corpus callosum. Which of the following is not part of the brainstem? So you're coming up the spinal cord and you hit the brainstem, which is composed of 3 parts. And the part that is not the brainstem is the thalamus. The first part you hit is the medulla oblongata, then the pons, and then the midbrain. Which part of the brain contains the cardiovascular and respiratory centers? Medulla oblongata. But really you can think of the brainstem as, some of these basic controls. Surrounding our brain and spinal cord are layers of connective tissue called meninges. You may have heard of meningitis. The outermost layer is very tough. It's called the tough mother, dura mater. The outer layer is fused to the bone's membrane. Remember, covering the bone is the periosteum. The arachnoid mater is the middle layer. It looks more like a meshwork. And the pia mater is a shiny, thin, delicate membrane right on the brain itself or spinal cord. These are the same for the brain and spinal cord. Here's some detail that you can skip, but this is about how the cerebrospinal fluid travels. Again, you can skip all this, but we make about a couple cups of cerebrospinal fluid every day. The brain is divided into lobes, and the lobes are named for the overlying bones, as I said earlier, the frontal, parietal, temporal, and occipital lobe. But there's a 5th lobe that is deeper, and we'll see where that is in a second. There are some landmarks on the brain that we need to know. Some sulci, the lateral sulcus, the central sulcus, and the precentral gyrus, postcentral gyrus. And I'll show you those in a second. Okay, so here we are. Again, this is the left side of the brain. This is the anterior of the brain. This is posterior. If you were to go in here between the temporal lobe, right here at this lateral sulcus, that's where the midbrain is, called the insula. Here's the occipital lobe, parietal lobe, and frontal lobe and temporal lobe. So right here, between the parietal lobe and the frontal lobe is what's called the central sulcus. And there's a very important gyrus behind it and in front of it. So this one that's in front, the red one, is called the precentral gyrus. So of course, the one that's behind the central sulcus is the postcentral gyrus. So the precentral gyrus contains the primary motor cortex. That is where the information leaves the brain to tell the body what to do. And the postcentral gyrus contains the primary sensory. So again, we'll look at that. This purple stripe here is sensory, and this one is motor. Just the cortex, that outer layer of gray matter.